these drugs. And because we lack the target, it has a very high therapeutic index, right? I mean, it can't be toxic to us if we don't possess the enzyme for it, right? So it's going to have very high toxicity. Now, there are different types of sulfa drugs as well, okay? And it makes sense because generally synthesis pathways in any cell is a multi-step process. It's not a single step, single enzyme carries it out, done, right? It doesn't work like that. You're going to have several steps, several intermediates, and then the final product. So we have two sulfa drugs that are shown here, the sulfonamides and trimethoprim. Both affect the synthesis of folic acid, but they're going to target different enzymes and different steps of the synthesis, right? So the sulfonamide is going to prevent the enzyme dihydrotoroic synthase that is going to convert PABA into dihydrotoroic acid. And you have the trimethoprim that inhibits a different enzyme, dihydrofolate reductase, that converts dihydrofolic acid into tetrahydrofolic acid. Okay, so you can have these drugs that target the same pathway but act on different steps of that pathway, and they work really well. So these could be really good synergistic drugs because together they're going to give you a much stronger, sustained result than individually, okay? And because you were targeting two different steps. And because the final product is involved in the nucleotide synthesis as well as amino acids, both things important for every type of metabolic pathway, right? I mean, you need ATP for everything, and that's the purine, right? So, and amino acids are required for synthesis of all types of protein. So these drugs tend to be, cidal. They end up killing the microbes. Okay, questions before I move on? Okay, then you have drugs that target nucleic acids, and because there is a lot of course, similarity in the structure of DNA and RNA and the replication process and transcription process and so on, you have to be looking for a selective target, okay, and that's not always achievable. But once you do find a target, right, then that target could be shared amongst most prokaryotes, especially bacteria, but not humans, so it tends to be a very broad spectrum drug. And the quinoline, which is shown here, nalidixic acid, was the original drug. You find it in in in, um, in quinone water, okay, that people drink, and it's a natural compound, and it can target enzymes that are involved in DNA, replication. Now the issue with, like I said, we share a lot of these pathways. Issue with these drugs is that they have low therapeutic index. So even when we do find a target, there still is a lot of overlap in the target, that you can't achieve that complete selective toxicity. There's always going to be so much more toxicity to the host cells as well. So again, just to reduce that toxicity, you can change the R group. So here is the R group of nalidixic acid that was the original quinolone drug that has been changed so that now you have the norfloxacin. And they're gonna target an enzyme like a gyrase or topoisomerase that helps with the unraveling or packaging of the DNA during, say, the replication process. When we talk about it, I'll point it out. But those are the enzymes that would be targeted. And so these enzymes and their targets have to be specific to bacteria because we have those enzymes as well. So by modifying the drugs, and hopefully combining it with a target that is at least somewhat more specific to bacteria, then the patients, then we can hopefully achieve slightly higher therapeutic index than with the naturally occurring forms. Okay, so there are some that will target those DNA enzymes involved in replication and maintenance of DNA. And then there are others that are involved at transcription. Transcription happens to be the toughest thing to target because there is very little difference in the processes between bacteria and eukaryotes in the patients, right? And so if you try to say target the RNA polymerase thinking, well, it only works at transcription, turns out that there aren't that many targets there that will selectively destroy bacterial polymerase, but not affect the host polymerases. Okay, so, you know, they can, of course, be um, still cidal because you can't make the RNA and you can't make the proteins, but they can have severe side effect on our cells as well. So these are again, tightly regulated. These drugs are only given as a last resort, especially if they have low therapeutic index. So just to give you another example, here is a drug called actinomycin D. Okay, and so here's the structure shown. I don't care whether you memorize it or not, but at some point I guess you will have to learn it, just not right now. But so actinomycin D will bind to the GC residues, and once you intercalate between the GC residues, it prevents the replication and transcription at that site. So unfortunately, it can do the same thing for our cells as well. So you can immediately see that it's not very selectively toxic. So this drug might work really well on a petri dish against bacteria, but we can't use it for treatment of infections because it's gonna be very very toxic to our cells as well. Our cells will die just as much as the bacterial cells will. Okay, so there's a problem with taking nucleic acid inhibitors. Rifampin is a drug that is used to treat um, TB infections, and it targets the RNA polymerase um, site, very, very specific site, where the newly synthesized RNA is emerging from an opening. Okay, that's how specific it is, that it binds to a place on the RNA polymerase that is only unique to these mycobacteria and not present in the host cell. So there's some uniqueness there that we're targeting. Now, rifampin is going to then target, of course, the transcription process, and it's going to eventually kill the cells. And it comes as an orange powder, so it's anybody who's taking it, it makes all of their body's fluid orange as well. So their tears turn orange, their urine is orange. If it's a lactating woman, her breast milk is going to be orange as well. So not very pleasant side effects. But obviously, people who have the infection, TB, they have very little choice when it comes to drugs for treatment. To give you another example, metronidazole is a drug that is used to uh, cleave DNA randomly. So I told you, well, actinomycin can do it, but it binds to GC, and we are not going to use it. But how can then you use another drug that cleaves the DNA and destroys it, right? I mean, wouldn't that do the same thing for our cells? So it turns out that this drug works a little bit differently, that this drug is taken in its inactive form, so prodrug form, so that it's not active, and it'll be taken up by cells, but only in the bacterial cells in presence of an enzyme, flavodoxin, would this become active. Okay, so that there's an electron involved, and you have the nitrous form uh, that is going to then cleave the DNA randomly. So this conversion to this free radical form is only going to happen in the bacterial cells and not in the patient's host cells. So it spares patient's DNA but destroys the bacterial DNA. Okay, so you have to, as you can see, study the drug, its effect both in uh, bacteria as well as in patients, and see how the drug might change, how it might become active or it might become inactive. Some drugs, such as the sulfa drug, originally when it was isolated, it was tested against bacteria in a petri dish. It didn't work against the bacteria, but the researcher who was working with them, he was so desperate because his daughter had a terrible infection, and he, in desperation, just inoculated her with the sulfa drug, and her condition was cured. Her infection was cleared. Okay, so you can have instances where drugs don't really work in a petri dish, but can work in a patient's body, or before then, of course, in some model organism's body. So keep those things in mind. Okay. Um, Question. Yeah, so metronidazole also works against protozoans. Yes, absolutely. It works against protozoans as well. I think I mentioned that. Okay, against both bacteria and protozoans, so Giardia and Amoeba, Trichomonas, all parasitic infections, Entamoeba and Giardia of, for example, intestinal tract by eating contaminating foods. Okay. Okay, then you have drugs that target, proteins synthesis. Okay, and there's so many different targets, right? Ribosomes, we talked about that bacteria have unique ribosomal proteins, right, that they don't necessarily share with us. So we want to target those particular spots that they don't share with us, because ribosomes we do share with them. And ribosomes, uh, that bacteria have are 70S, those that we have are 80S. And so, because all prokaryotes have 70S ribosomes, one drug that works against one, ribosome is going to be pretty broad spectrum, because all microbes have ribosomes, right? But those ribosomes can change the binding sites for these drugs, the, on the ribosomes, and not respond to these drugs, right? So then you have to individually study these drugs and see where exactly they act. And so, because they prevent protein synthesis, they don't right away kill the microbe. Microbes can survive until you continue to take the drug. You stop taking the drug, and they can, kind of emerge from this inactive state and actively begin to metabolize. So you generally will have to trust your immune system to go out and phagocytose and destroy these microbes as they are laying inactive. So there are different, drugs that carry, different targets. I'm giving you a few examples. You have the aminoglycosides that I'm sure you have taken at some point. Streptomycin, a lot of us take. Um, and when you look at the target of aminoglycosides, what we notice is that they basically prevent the ribosome from correctly gliding along the mRNA, okay, so that it reads the correct codons to produce the correct proteins. The result is that it produces inaccurate proteins, which just are not going to fold correctly, and their numbers are going to continue to build up in the cells, and they're not useful. It takes a while to degrade them and realize that there is a problem, and the cells would have, been, already irreversibly damaged by then. Then you have tetracyclines. The name is given because of the four-ring structure. Your aminoglycosides, their name is given because these are amino sugars that you have, and they have this cyclohexene ring that is specific to them. So you have the tetracyclines that have four-ring structure, and they bind to the A site, okay, on the ribosome, and they prevent the incoming tRNA to come in and bind. So ribosomes might assemble and begin the process of synthesis, but can't really complete it because at some point their A sites would be taken up by tetracyclines, and protein synthesis can't be completed. Then you have chloramphenicol that prevents the the peptidase enzyme, okay, that we refer to as the peptidyl-transferase, that is responsible for linking together amino acids. It prevents them, it prevents that enzyme from generating this peptide bond. Okay, you have the oxazolidinones that are gonna bind to the large subunit, and it's gonna prevent the large subunit to combine with the small subunit so that you could initiate the process of protein synthesis. You can have macrolides, okay, erythromycin. You at some point must have taken erythromycin. Um, it can bind to it's both the large and the small subunits, link them together. Okay, so with the mRNA, so as the ribosome tries to glide along the mRNA to read all the remaining codons, it can't do it. Okay, so it kind of is stuck in the position that it's in. So you have these various drugs that are going to target the translation process by targeting the ribosomes that are specific to components that are specific to bacteria. uh, so which one do you choose? You choose the one that is the least toxic, right? Um, chloramphenicol isn't prescribed a lot, especially to pregnant women or people who are immunocompromised. Um, you know, because it affects the normal rate of blood cell production. Because somebody who's taking it could have anemia for the duration of the drug, or could develop potentially life-threatening anemia. But it's good for meningitis infection because it has the ability to cross the blood-brain barrier and reach the tissue in the brain. So it depends on what your target bacterium is, what your target compartment in the body is, and what's the therapeutic index is, and so on. Okay, so there are various features that need to be looked at. So in terms of, I said that, well, you want to be careful with how much drug you're using, because you want to prevent these microbes from attaining this resistance to drugs. So, you know how does this come up, and is it naturally present in the environment? And so we tend to sort of um, associate this development of antibiotic resistance just to use of antibiotics. But the truth is, the microbes have always had antibiotic resistance genes. Using antibiotics kind of kills off bacteria that are, sensitive to the drugs, and only the resistant microbes survive, and they go on to reproduce and take on the population. So same thing happens when you start taking an antibiotic and you stop taking it in the middle of it, or you don't take it as prescribed, so that you take whatever you want, right? So your minimum inhibitory concentration is maintained. You would have some bacteria that are resistant. Those are the ones that are going to take the longest to die, right? Those are going to remain. If you stop taking the drug, then those bacteria grow, and these are the ones that are going to contribute to the larger population. And this can sometimes lead to superinfections, okay, where you have the accumulation of antibiotic resistant bacteria, because all the sensitive ones have died out, and these were kept in check by those sensitive bacteria. All right, so their numbers were reduced because they couldn't compete well with the non-drug resistant cells. Now you've killed off all the sensitive bacteria, and now it's given these microbes the chance to continue to grow and take over the population, and they start to give rise to infections that are associated with destruction of normal microbial flora, like antibiotic associated colitis due to Clostridium difficile infections, referred to as CD infection or C. diff, which is when these Clostridium microbes would begin to burrow into your intestinal lining, and cause profuse diarrhea and bleeding, and it can lead to death very, very quickly. Okay, so you have to be careful. This isn't something that you should take lightly. Why would microbes produce antibiotics and have genes to produce antibiotics? As I mentioned earlier on, that they probably want to have some sort of competitive edge. But in terms of how do they protect themselves, right? So if they're going to produce a antibiotic that targets a particular structure, they might have a similar structure. So how do they protect themselves? So one, they can either lack the target. So a mold produces penicillin, it doesn't have peptidoglycan and so the drug is only going to target bacteria, not the mold itself. It itself can produce also an enzyme that helps to destroy it within the cells if it is toxic to itself, and so it only will be effective in the external environment against the other microbes. It can be produced as a prodrug, release outside and become active only outside the cell, or and then once it's active outside, you want to prevent its entry by generating these pumps that are active in the outer membrane. So it comes in, you pump it right out. Or they can have modified targets, like they can change the ribosomal structures so that the ribosome targeting drugs don't bind them themselves. And so these microbes have then genes that also destroy the drug, and they can share these genes depending upon needs in the cell, or in the environment. But the major ways of sort of passing this genetic information that is generally carried on resistance plasmid between bacteria is referred to horizontal transfer, and there are three ways it can happen. One is that a cell that carried a drug resistant genes dies. It's fragmented, it releases its DNA into the environment, and some cell randomly picks up a piece of DNA that contains that resistance gene on it, and it incorporates it either into its genome or into a plasmid. Okay, and it keeps it because it doesn't know that it's going to be useful to it right now, but under pressure, it will realize its utility. So that's one. It's called transfer. Okay, transformation, transfer of free DNA. The other is where the cells can physically link together because they contain an F plasmid, fertility plasmid, that allows them to produce a sex phyllis and pass on genetic material through that, and they generally pass on resistance plasmids to share resistant genes. The third way is transduction, okay, which is when a virus may be filled with uh, um, bacterial DNA instead of viral DNA, and if that bacterial DNA carries a resistance gene on it, when the virus goes and infects the next host, it's going to inject that resistance gene into the next host instead of the viral genome, and so the next host is not going to undergo the virulent cycle for the phage. It's just going to essentially you know um, um, incorporate that, and then eventually just maintain it. Okay. Let me just finish these last two slides and then I'll stop. A lot of the hospital acquired infections are due to the microbes that are listed here that are responsible for a third of all the UTI infections and suture infections and pneumonia and sepsis and hospitalized patients. A lot of these microbes have acquired this drug resistance via one of these means, and so it's important to then detect if the microbes are able to resist some of these drugs. And so the way it's done is that we either look for the product, do they have a penicillinase, or can we detect the presence of the gene via PCR, for example, that codes for penicillinase. So we're going to talk about, PCR later. So the mechanisms of drug resistance, what do those, mechanisms mean? They're microbes that are going to have this intrinsic resistance, which is that they're going to have the gene or a modification to their structures that prevents the drug from acting upon them, or they can get it from the environment where they can acquire it from other bacteria via this horizontal gene transfer, or they can mutate certain structures that would prevent the drug from binding. So in terms of enzymes, they are always coded by genes. So you can just suddenly synthesize a gene. You acquire it from the environment, or you had an intrinsic ability where you always had a gene for that particular enzyme. So penicillinase, we talked about that it attacks the beta lactam ring and destroys it. Then microbes can activate um, um, pumps, okay, where the drug comes into the periplasmic space, for example, or inside the cell. and they turn on these pumps that are not selectively going to pump out one drug. They're going to pump out all different types of drugs. And we have those types of drugs in our cells too. A lot of time, pancreatic cancer, for example, is so deadly because the cells turn on these pumps that pump out all types of cancer drugs that we use to try to treat that cancer. Then you can have the receptors that are involved in internalizing these drugs. You looked at their structure is very hydrophilic, very large. They're not going to simply diffuse across the membrane. And, so there's going to be some sort of transport protein. Bacteria can change the transport protein via spontaneous mutations. And when you put them under pressure, which means you put them in presence of an antibiotic, they suddenly survive because they can't allow this drug to enter. You can have change in the binding pathways involved. Microbes can have multiple pathways that lead to the synthesis of the same product. And if using a drug that targets one pathway, microbes can abandon that particular pathway and start using an alternative pathway to, achieve the same effect. So this you have to have enzymes and genes corresponding to those enzymes and pathways. This could come up due to spontaneous mutations, right? Um, then similarly, activating a drug pump, you have to have genes corresponding to those pump. Decreased permeability could be due to spontaneous mutations, and drug inactivation, you again have to have genes corresponding to production of those enzymes. Okay, thank you so much. I'm going to stop here. Any questions?